Okay, so let's get started. Hello everyone. Welcome to our today's Zoom seminar. So it's my pleasure to welcome our today's speaker, Dr. Ni Jinfei from Fudan University. Jinfei currently is a assistant professor, also junior principal investigator in the Institute of Translational Brain Research at Fudan University. So Jinfei complete his undergraduate study in Fudan University, and then after that he moved to U.S. And then start his master program training at University of Connecticut Health Center. And then after which he received a PhD training in Johns Hopkins School of Medicine. So from 2017, after he finished his PhD training, he moved to Boston and then studied in Harvard Medical School, HHMI, as a postdoc fellow in Stephen Lieber's lab, who is also a pioneer in Vegas neurobiology. And then from 2021, Jinfei complete his postdoc training and then moved back to China to Fudan start his independent research program. The main theme of Jinfei's lab is to study functional dissection of the enteric nervous system and gut-brain neuronal connection, which is also a big interest of my labs as well. So the interest in Jinfei's lab is to study the gut as an organ responsible for food digestion and absorption, but also at the same time the GI tract function as a body endocrine and immune organs. So his study trying to integrate both the nervous system and immune system together, and then to study the molecular mechanism between these organ-organ interactions. So different systems also can be found in the GI, work together to maintain the metabolic regulation and the immune defense. And then, as I just mentioned, the GI tract have multiple functions which can be controlled by the nervous system. Also includes particularly includes the extrinsic nervous system, which including sympathetic and then parasympathetic system, and also including the enteric nervous system. So his group used mice model and also same time employee multi-omics analysis and then different viral label technologies and neurophysiological method imaging approaches to study key questions related to the development and the functional modules of gut neurobiology and the gut-brain connectivity. The study conducted in Jinfei's lab focused on these systems function in metabolic regulation and immune defense. In addition, also analyze clinical patient samples aimed to further explore the neurobiological mechanisms underlying human gut-related disease and develop target therapeutic strategies and drug interventions. Really exciting to have Jinfei with us today. And then I think he's also going to talk about the gut interception and also GI physiology. So welcome, Jinfei, to our today's talk. Very much looking forward to it.
Okay, thanks, Chen, for this nice introduction. I didn't realize that Chen get so many information from my potential laboratories interest that I haven't even put out there on the internet. And if I know you are very enthusiastic about the enteric nervous system, I would prepare a different slides. But today's talk is mostly talk about nervous system that resides out of the gut, that regulates the GI function. So it's really my great pleasure to share with you two pieces of our unpublished work on the topic of autonomic regulation of the physiology, of the gastrointestinal physiology. As the commander-in-chief of the body, the brain relies on a fascinating and critical bidirectional communication system with the internal organs to ensure our body functions seamlessly. This communication involves both sensory and motor pathways. The sensory pathway here, denoted in red, inform the brain about the state of the internal organs. And the brain then integrates these sensory information and send out motor commands to regulate organ physiology. The autonomic nervous system is the key player in brain-organ communication. It operates unconsciously and has two main branches. The sympathetic nervous system, often called the fight and flight system, prepares the body for action by increasing heart rate, dilating pupils, and redirecting blood flows to muscles. The parasympathetic system, on the other hand, known as the rest and digest system, promotes relaxation, slows the heart rate, and enhances digestion. Together, these systems ensure our organs respond appropriately to internal and external stimuli, maintaining body homeostasis. Our lab has been working on a bidirectional communication between the gut and the brain, with primary focus on organization principle of the gut-associated nervous system and how the gut physiology, including digestion, metabolism, gut immunity, and are regulated by the nervous system. Beyond the autonomic neurons located in the peripheral ganglion, the gut is uniquely equipped with its own intrinsic nervous system, the enteric nervous system. The enteric nervous system spans the entire gastrointestinal tract from esophagus all the way to the anus. It consists of neurons and associated enteric glia, organized into two primary neuron plexus, the myenteric plexus and the submucosa plexus. The submucosa plexus is located beneath the gut mucosa, plays a key role in controlling secretion and blood flow, while the myenteric plexus is located between two layers of smooth muscles in the muscle wall and are primarily functions in regulating gut motility. Often referred as the second brain or the brain of the gut, the enteric nervous system forms a highly organized neuronal network composed of diverse neuronal types. This network is essential for nearly all gastrointestinal functions, including digestion, absorption, motility, gut blood flow, endocrine, and metabolism. And therefore, it evolves in many types of gut-associated diseases. Remarkably, the enteric nervous system can operate independently without requiring input from central and or the autonomic nervous system. Nevertheless, the enteric nervous system is also closely associated with the rest of the autonomic nervous system. For instance, in this tracing experiment conducted in our lab, we injected AAVs express two different fluorescence proteins into the dorsal motor nucleus of the vagus and the celiac supramesenteric ganglion. By the way, can you see my cursor on the screen? Okay.
Yeah, we can.
Okay. And to label both the parasympathetic terminals and the sympathetic terminals within the gut wall. And often we can see that the terminals from the sympathetic and parasympathetic systems are closely associated with the enteric nervous system labeled in blue. On the other hand, both the vagal-derived and the dorsal root ganglion neuron-derived sensory terminals are also often located close to these enteric neurons in the gut wall. These histological analysis revealed that the terminals of both the motor pathway and the sensory pathway of autonomic nervous system is closely associated with the intrinsic nervous system, indicating that the enteric nervous system plays a key role in even in communicating the brain with the gut. So the first story I would like to share today is regarding how the sympathetic system, one of the very important motor output system, is organized into functional units for regulating organ functions. So the sympathetic system plays a critical role in fight and flight response. When faced with danger, it triggers physiological changes such as increasing heart rate, deepening breath, slowed gastrointestinal motility, and constriction of visceral blood flows. These changes play a role in redirect nutrients and oxygens into critical organs like the skeletal muscle and the brain for fight and flight response. Historically, this sympathetic system was thought to act in a bundled all or none manner, which is why it's named sympathetic system, reflecting the idea of a unified response across the body, like a conductor of the symphony. However, growing evidence reveals that the sympathetic system also plays indispensable role in maintaining organ-specific homeostasis. For example, in this baroreflex pathways, the sympathetic activity adjusts the heart rate and vascular tune, but without significant changes to gastrointestinal motility or the metabolic organ function. This suggests that the sympathetic system operates with a higher degree of specificity than previously thought, enabling fine-tuned organ physiology regulation and maintain overall physiological homeostasis. Accordingly, the sympathetic system has been proposed to carry out its diverse functions through distinct functional units, such as the cardiorespiratory units to regulate heart rate, lipolysis unit to regulate metabolism, visceral motor units to regulate gut motility, and immune regulatory units to regulate immune function, and visceral constrictor and circulatory motor units to regulate blood flow and secretion. And it's the permutation and combination of different functional units is implemented when the body faces different disturbance of the internal homeostasis. But are there specific sympathetic units to carry out these sympathetic functional units? How these sympathetic units are organized is what we are interested to address. To address these questions, we first take a look at the overall organization of the sympathetic system in the peripheral. The sympathetic nervous system is composed of two groups of neurons organized in series. The presympathetic ganglion located in the spinal cord and the post-sympathetic ganglion located in the peripheral ganglion. And it is we start our analysis in the postganglionic neurons because these neurons direct facing the internal organs. They are the final motor pathway of the sympathetic system. And for the post-sympathetic neurons are located in ganglions across the body in multiple ganglions. The post-sympathetic ganglion we are interested in study is called celiac supramesenteric ganglion. It's located in abdominal and supplies the majority of the sympathetic outflow to various types of abdominal organs. So that's why we focused on this particular sympathetic functions. And previously, regarding previous physiological analysis indicates that within this sympathetic ganglion, there are at least 3 functional units supply the GI tract. Visceral motor units that most likely targeting the smooth muscle layer to regulate gut motility. Circulatory motor units most likely targeting the submucosal layer to regulate the gland secretion in the mucosa. And vasoconstrictor units to constrict the blood vessels in the GI tract. But are they carried out by different types of sympathetic neurons, or they are carried out by similar sympathetic units, and their functional specificity is determined by where they are targeting. To answer these questions, we take three different approaches. First, we using single-cell transcriptomics to chart and single-cell atlas for the celiac superior mesenchymal ganglion. And next, we analyze their projection morphologies of genetically defined sympathetic neurons within this ganglion. And third, we're using various types of genetic tools to modulate these activities of these neurons to look at how they regulate gut physiology. So first, we are using single-cell sequencing to really dissect the diversity within this ganglion. And as you can see that within this sympathetic ganglion, there is not only sympathetic neurons, but also Schwann cell-like sympathetic glias and other types of supporting cells. By using sympathetic neuron-specific markers such as tyrosine hydroxylase, we subset these neurons and then reclassify them into 15 different subtypes. For many of the subtypes, we can identify unique markers, which we use them to generate myosin to hope for labeling different subtypes of neurons. And then we using AAV injections to infect specifically these CGSMG ganglions to express fluorescence proteins and look at their terminal morphology across different abdominal organs. Here is a verification of the TH staining, co-staining with the tomato-positive fibers in the gut wall, indicating that we are indeed labeling specifically these sympathetic neurons, and we analyze these sympathetic terminals. So our analysis in the GI tract revealed that multiple terminal types exist from these sympathetic neurons. In the myenteric layer, in the muscle layer, we mainly observe two types of terminals. Terminals that located in the smooth muscle layer that forms array-like shape called intramuscular arrays. And there is terminals formed surrounding the enteric ganglions labeled in green. We call intraganglionic basket endings. These endings are very wrapping around, as you can see here, these enteric neurons and enteric glias. In the submucosa layer, we find another types of endings that form this tube-like structure, which we call perivascular endings, because these tube-like structures are like vessels. And on the other hand, we also find terminals that forms basket endings surrounding the enteric ganglions in the submucosal layers. So to further dissect what are these perivascular endings are, whether they are associated with blood vessels or associated with lymphatic vessels, we performed a co-staining with the sympathetic terminals using the fluorescence protein, and then the CD31 to label the blood vessels, and the LIF1 to label the with the lymphatic vessels. As you can see, these perivascular endings only exist in the submucosal plexus, and they are closely associated with blood vessels, but never associated with these lymphatic vessels. As you can see here, there's a labeled perivascular endings, but they are not associated with the blue labeled lymphatic vessels, but they are associated with here CD31 associated blood vessels. So in summary, our morphological analysis indicated that sympathetic neurons form at least three types of endings: the intraganglionic basket endings associated with enteric neurons and enteric glias, perivascular endings that associated with blood vessels, and intramuscular endings that exist in the smooth muscle layer. So next, we dive into different types of reporters we obtained through the single-cell analysis and look at whether different subtypes of enteric neurons form different terminal morphologies. And here we analyzed, among the crylines we analyzed, there's two crylines that give us the most exciting results. The cry that labels class 4 neurons, when we analyze them, we find that they only innervate the smooth muscle layer of the GI tract. By the way, when we do the overall sympathetic ganglion morphology analysis, we also find the terminals within other abdominal organs, including the pancreas, the spleen, the gallbladder, and the liver, and even the mesenteric lymph node. But here, this class 4 neurons labeled by this gray line only form terminals in the GI tract and only form terminals in the muscular layer of the GI tract, as you can show here. And the terminals are often form these basket endings surrounding the enteric ganglions in the muscle layer. And they don't form terminals here in, for example, in the pancreas eyelid. So this makes us think that this possibly function as the visceromotor units, because visceromotor units regulate the motility of the GI tract, and the motility of the GI tract is regulated by the smooth muscle layer neuron plexus. And the other class 12 cell types labeled by these gray lines form another different morphology types. We can see here that in the gastrointestinal tract, this class 12 neurons only forms perivascular endings, but never forms endings that surrounding the anterior ganglions. And for this particular group of neurons, they also form terminals in other abdominal organs, including the pancreas, the spleen, the liver, and in every organ we examined, they all form terminals associated with blood vessels. So therefore, we think that this class 12 neurons function as vasoconstrictor neurons, that they regulate the blood vessel tune. So by our single cell analysis and class-specific gray-line morphological analysis, we think that we find two group of neurons that morphologically indicating the place role as vasculomotor neurons and vasoconstrictor neurons. So next, we want to test if they indeed play a role as vasomotor units and vasoconstrictor units. We using chemogenetic approach to express chemogenetic activation units in these vasomotor units and vasoconstrictor units individually. And to analyze the gut motilities, we gavaged animal with common rat, and then measured how long it takes for the rat food to form the first fecal pellet and exposed by the animal, and measured as the total GI transit time. As you can see here, when we activate the vasomotor units with this chemogenetic approach, the total GI transit time is dramatically increased compared to the control units or compared to when we activate the vasoconstrictor units. It's similar to the similar delay was observed when we overall activated the sympathetic ganglion. And the number of faces exposed by the animal within the 5 hours is also drastically reduced. This results indicating that indeed these vasculomotor units not only morphologically adopt this unique structure to regulate motility, but functionally indeed works as a vasculomotor units. To measure the blood flows, we're using this laser spectral meter to look at the blood flow within the blood vessels of the GI tract, and using optogenetics to activate different groups of sympathetic neurons. And as you can see, when we shine the light, the blood flow starts to reduce, indicating a vasoconstriction. And here is the result of a trace. When we shine 3 different light pulses separately in control animals, it did not affect the blood flow at all, as you can see here. While we activate the vasoconstrictor neurons, each time we observed a reduction of blood flow, tightly linked to the light, shining light. And when we activate the vasculomotor units that regulate the motility of the gut, there's no change of the blood flow. So this experiment indicates that indeed these perivascular endings function as vasoconstrictor units to regulate the blood flow of the. Here we tested the GI tract, but I suppose it's similar in other abdominal organs. So in summary of our sympathetic work that we already submitted, we draw an atlas of the celiac sympathetic ganglion, which I did not expand my presentation here, but we analyze in different developmental times, and we analyze sympathetic neurons targeting different organs. We analyze their projection specificities and terminals within the various types of abdominal organs with a focus in the gastrointestinal tract of different subtypes of sympathetic neurons. And from this analysis, we find two separate groups of neurons. One is class 14, class 4, that from morphology and function analysis indicating that these are the visceral motor units that regulate gut motility only. And the other classes that form perivascular endings, they do not regulate gut motility, but they regulate the blood flow of the GI tract. From this work, we think that different sympathetic functional units are really encoded, at least in the post-ganglionic neuron level, by genetically defined separate cohorts of neurons. And this dissection, keep in mind that this is the last layer of the whole sympathetic pathway that facing the organs directly. From that dissection, we think that sympathetic functional units in the last interface between the sympathetic system and organs are encoded by genetically defined units, and this really serves as an entering point using tracer tools in the future to look at how brain really implement these different sympathetic functional units, whether there exists convergence, that exists neurons that can coordinate these functional units in the same times, or there are separate pathways as label lines to regulate these functional units. And these would be our future studies. So the second story I would like to share is regarding the sensory pathways for organ-brain communication. As I introduced, the sensory pathways inform the brains about the state of the organs, and it provides critical feedbacks for brain to regulate the organ physiology. There are mainly two types of sensory pathways in the peripheral nervous system. The vagal sensory pathway with its sensory neurons located in the nodostegular ganglion in the neck area, and the spinal pathway here labeled in greens with its sensory neurons located in different dorsal root ganglions located from rostral-caudal levels. The vagals for the interoception, for the brain to sense the state of the internal organs, the vagal pathway has been most of the attention in the last decades, with many publications indicating that the vagal sensory neurons plays a role in sensing sensory information from various areas of the body, including the laryngeal, the blood vessels, the lung, the stomach, for mechano and nutrient detection, and even for tissue inflammation. And often, the vagal sensory terminals, when they reach individual organs to sense organ-related sensory information, they are associated with local cell types. For example, here associated with keratic bodies in the keratic organ to sense blood gas, and associated with the neuroendocrine bodies within the lung to sense lung-related stretch and chemical information, and associated with the taste bud in the laryngeal epithelials to sense the chemical information in the epithelia. So this would become a theme for many of the interoception pathway. So, however, the spinal pathway hasn't really attracted much attention for its function in interoception. So the spinal pathway are known to mediate many of our somatic sensations, including touch, pain, itch, and temperature. But in fact, there's about 5% of the sensory neurons from the DRG from the dorsal root ganglia innervates not the skin, but the internal organs, and plays a role in relaying sensory information from organ to the brain. To analyze this spinal pathway, we first, there's recent publications about the mechanosensory channels, piezo, in mediating in the dorsal root ganglion to mediate organ-related interoceptive pathways. However, to gain a comprehensive view of how the dorsal root ganglion, the spinal sensory pathway, innervate various organs, we adopted an intersectional genetic approach to specifically label the spinal sensory pathways. In this, we adopted a Cray-on flip-off reporter, which will label a Cray-specific, which will drive a Cray-specific fluorescence protein expression. But when combined with a flippo line, the fluorescence reporter will get shut off. And the flip line we use is FOX2B flip. And this flip line labels most of the peripheral nervous system, but not the dorsal root ganglion. So this really provides us an opportunity to exclude the neural terminals from all the other peripheral organs, like peripheral ganglions, like the sympathetic ganglion, parasympathetic ganglion, the enteric system, and the nodal and the vagosensory neurons. And when we combined with VGluT2 which labels all the sensory neurons, we can see that the dorsal root ganglions get labeled, but the nodal ganglions are not get labeled at all. The jugular ganglions also get labeled, but they only innervate the head and neck area. So then we're using this approach, we find widespread DRG afferents in multiple visceral organs, including the GI tract, but also the trachea, the esophagus, and all these other organs. We dive in to form a deep quantitative analysis within the GI tract and find that the DRG, the spinal afferents are enriched in the distal part of the GI tract as previously reported, but also have substantial innervation in the stomach and the rest and the small intestine, although the density in the small intestine is much lower. Next, we're using optogenetic approaches to specifically activate these sensory terminals within the GI tract, within the colon, because we can stick an optical fiber in the colon, and to see what kind of behavioral response these DRG sensory pathway will induce. And we find that activating the colon sensory neurons induce the stereotyped nociceptive reflex, which the animals really hunch their back and lift their abdomens, which is indicative of a nociceptive sensation. And here is the quantification when we activate these neurons with optogenetics, the frequencies of this back hunching event is drastically increased. And now the visceral pain sensation is not only can induce this reflective behavior, but also associated with negative emotions. To test if activating these DRG terminals in the colon is indeed induced visceral pain, we test its negative emotion, whether it can induce negative emotion. So here we perform a step-down assay, which the animal was put into a platform with around 1 centimeter high, and with its optical fibers stuck in its colon. And when you lift the barrier, the animal will explore and quickly step to the floor of the behavior chamber usually within half a minute. And when they step to the floor, we deliver optogenetic activation. So the animal will couple the floor with whatever sensation they receive that caused by the sensory neurons in the colon. And we repeatedly doing this. And if the optogenetic activation is induced a negative emotion, then in the next round, the animal will not like we're reluctant to step down from the platform. So as you can see here, we quantify how long it takes for the animal to, from the platform to the floor as a step down time. As you can see from the green bar, which we put a balloon to extend the colon of the animal. So it takes the animal longer and longer time to step down to the floor because they know that when they step to the floor, they will receive a stimulation that is unpleasant. And when we do in optogenetics, we also observe the similar delay of the step down time, indicating that indeed optogenetically activating the DRG terminals in the colon will cause the negative valence. So here we're using intersectional genetic method to reveal the extensive DRG afferents in multiple visceral organs. And we analyze different subtypes of DRG, which I would not dive in details in the colon that can mediate different degrees of nociceptive reflex. And these sensations are aversive, indicated by our step-down assays. So when we analyze the terminals of the spinal sensory neurons, we often find that, as I mentioned, that they form structures associated with intrinsic nervous system, the enteric nervous system. As you can see here, from all areas of the GI tract, they always, many of them forms this, endings associated with the enteric ganglions. And this makes us very think that whether there's a putative functional coupling between the intrinsic nervous system, the enteric neurons, and the spinal sensory pathway. Because the enteric neurons from others and our analysis also contain sensory neurons. And if the external sensory fibers are associated with the enteric ganglions, whether the enteric neurons can relay sensory information through the DRG spinal cord to the brain to induce behavior. Because if that's true, it will, it will change our understanding of the enteric system. The previous understanding think the enteric nervous systems only relay its information within the gut to regulate gut physiology, would not cause any consciousness or behavior changes. So to test this, we inject AAV locally in the colon to express channel rhodopsin. And this method will very specifically only labels the local enteric neurons. And in the same approach, we're using optogenetics to activate these neurons in live animals. And first, we look at C-force expression in spinal cord dorsal horn. And we find that there's an increased C-force positive neurons in the dorsal horn area of the spinal cord, indicating that activating the enteric neurons with optogenetics indeed induce neuron activation of the spinal cord. Behavior analysis indicating that indeed activating enteric neurons can induce this back hunching reflex, similar to what we observed when we activated the sensory terminals from the DRG directly. However, when we're using the step-down assay to analyze the emotion associated with activating enteric neurons, there's no delay of the step-down time, indicating that in normal and under normal conditions, activating enteric neurons induced a behavior reflexes, but the sensation is not very aversive. That is different from activating the DRG terminals in the colon. And so to further test whether the behavior response we observed from activating enteric neurons is indeed through the spinal pathway, we're using RTX to ablate nociceptor DRG neurons. And when we activated enteric neurons in control animals, we can induce strong back hunching responses. But when we ablated the DRG neurons while activating these enteric neurons, we did not induce any back hunching response, indicating that indeed the behavior response we observed is through the spinal pathway. So we find that the colon enteric neurons can induce nociceptive reflex through the DRG spinal pathway with minimal level of aversion. So the enteric neurons are composed of many different types of neurons that that compose the sensory neurons, motor neurons, and interneurons. And what type of enteric neurons are mediating this enteric DRG pathways? To test this, we subdivided enteric neurons into two major subtypes: cholinergic subtypes labeled by Chattree and non-cholinergic subtypes. We find that activating cholinergic subtypes can induce this nociceptive back-hunching response, also induce a drastic increase of the spinal cord dorsal horn cephus expression. And on the other side, when we knock out Chat gene in these enteric neurons, optogenetic activation no longer can induce either behavior response or the cephus expression, indicating that cholinergic neurons and the acetylcholine they release can mediate enteric neuron DRG signaling. On the other side, when we activate non-cholinergic neurons in the gut using VIP-CRE, we no longer observe the strong batch hunch expression. And the cephus induced by activating the VIP enteric neurons is also much weaker than activating the cholinergic pathways, indicating that the non-cholinergic enteric neurons do not communicate with the DRG spinal pathway for back hunching response. Within the cholinergic subtypes, we put our investigation into these intrinsic primary afferent neurons, which are the primary sensory neurons in the enteric system. We want to test whether these IPENs are mediating this communication between the ENS and the DRG pathway. And indeed, with this Advilling-Filippo lines, we express optogenetic tools specifically in these IPENs in the colon, and we can drive strong back hunching response. And within these IPENs, we're using piezo-2 to activate these mechanosensitive enteric neurons. And indeed, the mechanosensitive enteric neurons are participating in this enteric neuron and DRG communication. Now, previously I mentioned that one difference between directly activating the DRG terminals in the colon and activating this indirect ENS DRG pathway is that activating the DRG pathway can induce aversion. The animal doesn't like this feeling. While activating the enteric neurons, although the animals make a behavior reflex, but they are not bothered by this sensation. However, as we know that under colitis inflammation, our visceral sensation in the GI tract is hypersensitized. So at the end, the patient usually feel much drastic levels of pain in the GI tract. So we wonder if colitis can change the sensations induced by the enteric system from non-aversive to aversive. So we use DSS to induce the colitis model, which is a mild level, which we feed the animal with 3% colitis for 5 days, and then change the water into regular water for the animal to recover from its colitis, and use the step-down assay. We find that in physiological conditions, activating enteric neurons do not induce aversion, as indicated that there's no increase of step-down time drastically in the blue bar, while in the acute phase of colitis, when the animal was under inflammation, when we activate the enteric neurons, the animal starts to feel these aversive emotions. And this aversion even persisted when the animal was fully recovered, at least from the body weight index, fully recovered from the acute colitis. However, when you activate the enteric neurons, the animal still feel negative emotions. And this is probably consistent with some of the clinical observations that even the patients was recovered from acute inflammation in the GI tract, their GI tract become hypersensitized, and they often feel much higher level of uncomfortableness, even when there's no acute inflammation in the GI tract. So in summary, our analysis developed a tools for dissecting spinal pathways for interception. We demonstrated an enteric DRG pathway to mediate gut interception, and this is most likely to be carried out by chorinergic enteric neurons through acetylcholine as a mediator. And we pinpoint a possible targets for inflammation-sensitized visceral pains. And it's possible that when the patient was suffering from inflammation, the enteric system get sensitized, and the enteric system's inflammation can induce an aversiveness that is in normal conditions are not aversion. And that's in summary, our lab focused on the brain-gut communication mediated by the peripheral sensory and motor pathways with an emphasis on autonomical neurobiology and interoceptions. And today I haven't had time to talk about how these gut-associated autonomic neurons regulate metabolism and immunity, but I hope in the future I can share you guys with stories related with these topics. And thank you very much.